Hello all, in this video we are going to discuss about indicators of malaria. In the previous video we have seen indicators of filaria. Now we are going to see the indicators of malaria. We have many indicators, that is during the pre-eradication era. We have used spleen rate, we have used these parameters, and there are certain vector indices that is given here. We all know malaria is transmitted by mosquito, that too Anopheles mosquito, transmitting the Plasmodium species of the parasite. So that we all know, out of which falciparum causes more dangerous. So we have indicators separately for falciparum alone. So that is the basic thing which you need to understand before going to the malaria indicators problems. In the pre-eradication area, we use this spleen rate, which is the proportion of children between 2 to 10 years of age with spleen increased size. So that is called as spleen rate. And the average enlarged spleen means out of this 2 to 10 years of age of children, the average size of the enlarged spleen will be measured as the indicator. And parasite rate among the children with 2 to 10 years of age, how many have active parasite, that is Plasmodium, in their blood using the thick smear of blood examination. So that also was used. Then we have the parasite density index, that is of the positive slides, how many parasites are present will be plotted as parasite density index. Then we have infant parasite rate, that is among less than 1 year infants, how many infants are showing positive Plasmodium in their blood, that is the infant parasite rate. And proportional case rate, this is out of the total cases in hospital admitted, how many are due to malaria. So that is a crude indicator which was used earlier for identifying the burden of the malarial disease. So these were the indicators which is not of use right now, and the chance of these indicators in exams is also very less. And we have vector indicators such as human blood index, that is out of the Anopheles mosquito, we have to take a set of Anopheles mosquitoes, and out of which how many have the human blood in their stomach will be considered as human blood index. It is a sign of anthrophilism, that is mostly the Anopheles feed on other animals also. So how much the blood is getting fed by the humans is an indicator. Human is measured in this indicator. Then we have sporocyte index. This indicator measures the proportion of mosquitoes having human blood, and here it is the proportion of the mosquitoes having Plasmodium sporocyte in their salivary glands is called as sporocyte rate. Then we have mosquito density rate, that is with one catch over time, how many mosquitoes are collected by the individuals. It is calculated based on the number of mosquitoes biting the humans. Then we have inoculation rate. This is the multiplication of the sporocyte rate and the man biting rate. So when we combine these two, we will get the inoculation rate. So this pre-eradication era indicators and the vector indicators are for theoretical purposes. Now in practicals and in reality for program purpose, we use these indicators, that is during this eradication era. So that those are the 5 indicators. I have arranged these 5 indicators in a specific manner for us to remember it easily. So that is the number 1 is the annual blood examination rate, slide positivity rate. This is otherwise called as ABER. This is otherwise called as SPR. This is SFR or slide falciparum rate. This is annual parasite index or API. This is annual falciparum index or AFI. Now we need to understand, I have arranged these parameters in a specific order. So I will explain what it is. So annual blood examination rate is, as the name indicates, it is an indicator of the blood examined in the year. So it contains number of slides examined in the numerator out of the total population. So it will be expressed in percentage. So usually they recommend 10% of ABER for effective malarial survey. So that is the 10% of ABER. We have numerator as number of slides examined divided by total population for ABER. Now slide positivity rate is out of the total slides examined, how many are positive is called as slide positivity rate. So the numerator will be number of positive slides for malaria divided by total number of slides examined, and it is expressed in percentage. So multiplication factor will be 100. So the same way slide like how we have slide positivity rate, we have slide falciparum rate. So instead of total number of slides positive for malaria, we have to substitute slides positive for Plasmodium falciparum in the numerator. Rest all remains the same, divided by total number of slides examined into the multiplication factor to receive the percentage. So we get slide falciparum rate. Then the fourth indicator is annual parasite incident. This indicator combines this ABER and SPR, that is annual blood examination rate and slide positivity rate. So we have confirmed cases during 1 year in the numerator divided by population. So population under surveillance, the multiplication factor here is 1000. The significance of this APA is it is the most important indicator used in program purpose. When this APA is greater than 2, then it will be indicated as high risk area for any state or district. So the malaria control measures will be more rigorous in those areas. So that is the APA having the population as the denominator and confirmed cases in the during the 1 year as the numerator. So if we replace the confirmed cases of malaria with the confirmed cases of falciparum, we receive the annual falciparum index. The multiplication factor, the denominator will be population under surveillance. So we have 5 indicators, actually 3 indicators, that is ABER, slide positivity rate and the API only. So since we have incorporated the falciparum in slide positivity and the annual parasite incident, the indicators have become 5. Annual blood examination rate, out of the total population, how many have received the blood examination is annual blood examination rate. And out of the blood examined, how many are positive, slide positivity. And out of the blood examined, how many are falciparum positive is called as slide falciparum rate. And out of the total population under survey, how many are positive for the case with a multiplication factor of 1000 will give annual parasite incident. And the same way, annual falciparum incident is calculated. Now we have this question here, that is the surveillance of malaria was done in a population of 40,000 in the year 2018. So the total number of fever cases given here is 10,000. Plasmodium vivax case is 400. Falciparum case is 600. Slides examined is 5,000. Now we need to calculate the parameters here, that is ABER, SPR, SFR, API and AFI. So I have arranged the question here. Then answer key is here. We will work it out. For ABER, the denominator is total population, that is 40,000 into 100. The numerator is number of slides examined. So that is 5,000. So if we do the mathematics, we get 12.5%. Then the next indicator is slide positivity rate. Again, here denominator is total slide examined, that is 5,000. And it is expressed in percentage. So multiplication factor is 100. The numerator is the total number of cases positive. So we need to add these two. So it will be 400 plus 600. So it will give 1000 as cases. So when we calculate this, we get 20% of slide positivity rate. Then we have the next indicator is slide falciparum rate. So for that, the denominator is number of slides examined, that is 5,000. Multiplication factor is 100. The numerator will be, so that is 600. So based on this, it will be calculated as 12%. Then the next important indicator is, that is annual parasite incidence. So we have to get confirmed cases. So confirmed cases here is 1000 divided by the denominator here is the total population under survey. So that is 40,000 into 100. So we get 25 cases, 1000 population per year. We need to write the unit also correctly. Then we have AFI, that is annual falciparum index. For that, the numerator is from confirmed falciparum cases, that is 600 divided by total population under survey into 1000. So multiplication factor here is 1000. So which will yield 15 cases per 1000 population per year. So that is the indicator of this annual falciparum. So there are 5 indicators. These two are similar and these two are similar. Here annual blood examination rate is the number of blood examined of the total population. And these 3 parameters are expressed in percentage. API and AFI will be expressed in per 1000 cases per population per year we will express. So the next is the slide positivity rate. Here the slide positivity rate is calculated by the number of confirmed cases divided by number of slides examined. Same way, number of falsified cases divided by examined. Number of confirmed cases divided by total population is API. AFI is number of confirmed falciparum cases divided by total population. So we have seen the indicators of malaria. Hopefully this video was helpful to you. If you like this video, please click on the like button and share it to your friends. If you haven't subscribed to the channel, please subscribe. Thanks for watching.